So thank you to Associate Professor Graeme Zosky for presenting today at the CAR webinar. Graeme's an Associate Professor in the School of Medicine at the University of Tasmania, and today he'll be presenting on can experimental models be used to answer public health questions, toxicology and beyond. I hand over to Graeme. Thank you.
Thanks, Amanda, and hello everyone. I'm sure you heard our discussion. I'd be more than happy for people to ask questions as we went. I'd rather this be a sort of learning process for both of us. The way that I've set this up is kind of in acknowledgement of the fact that in some ways I'm the token basic scientist in CAR, and so what I wanted to do was to sort of talk to people about what experimental models can and can't do in terms of public health. So the presentation is largely made up of 2 case studies, and in those case studies I'll sort of work through some of the work that we've been doing and how that fits into the public health framework, and how my approach to using experimental models to answer public health questions in a way. Because in the field in general, it's actually not, experimental models aren't used that often to answer public health questions directly. It's more common to use them for teasing apart molecular mechanisms and designing new drugs and therapeutics and all those sorts of things. And so there's a lot of my work sort of sits between that and the public health space. And so I just wanted to talk you through that a bit. So as a sort of starting point, I thought I would start just by, for those of you who aren't aware what's possible, just outlining what experimental models are, or what I consider experimental models to be. And there are sort of 3 broad categories. There's in vivo models, in vitro models, and in silico models. And in vivo models are basically animal models. And so the classic go-to animals, at least in the health space, are mice and rats, but there are certainly others. There are large animal models of disease. There are small animal models of disease. A lot of toxicological work, for example, is done on invertebrates and single-celled organisms. But in vivo models, I suppose the main advantage is that you have an intact biological system. So if the thing that you're trying to model involves interaction between cell types in situ, and you can't do the corresponding experiments in humans, then the in vivo models are sort of the closest you can get where everything is intact. In vitro models are usually cell models, and those cell models can be cell lines. And so a cell line is basically an immortalized cell line where you know that every cell that you grow in your culture system will be exactly the same every time. And so there are some cell lines that have been around for decades that have been used in innumerable studies. You can also use primary cells, and so a primary cell is where you take a cell from a human or an animal model and you actually study the behavior of those cells. The advantage of those models is that they allow you to isolate the mechanism. So, for example, thinking about the kinds of things that we're interested in, if you expose an animal model or a human in a human exposure experiment to a particle, you see a response, you might be interested in what's driving that response. And so cell models can allow you to unpack that. So what is the cell that's causing the response that you observe in vivo? And sort of the other end that's probably not talked about as much and is probably less relevant to the public health space are the in silico models. And so in silico models allow you to mathematically model complex biological systems. So the picture that I put up there is actually an example where someone has used basically gaming theory to try and understand the relationship between inflammation in the airway and what causes that airway to constrict in asthma. So the silico models can be really powerful because you can look at predictions in complex systems based on what you know about the biology. And so those are the 3 broad categories, and I suppose there is general pros and cons to each of them, but the overall pro to these systems is that they allow you to probe and manipulate them. So you can go beyond what you can do in a human exposure experiment, for example, to actually unpack and dissect the molecular mechanisms that are being affected. And you can also manipulate the system. And these days with gene editing, so I'm sure you've heard the buzzword of CRISPR being thrown about these days. These days you can knock out individual genes and individual cell types in an individual mouse, for example. And so you can modify the genetic programming so you can really isolate the effects. So you can knock out one particular pathway and determine whether or not that pathway is critical in the thing that you're interested in. And so that's the big pro. That's why they're useful, for example, in trying to identify new therapeutic targets, new drug targets, and so on and so forth. The big con, of course, is that you're far away from the human system that you're trying to model. And so even at the in vivo level, we know that there are fundamental differences between animal models and human physiology. The classic examples in the sort of response to particulate world are that mice, for example, are obligate nose breathers. So if you have an exposure chamber where you're exposing a mouse to a particle, those particles are going to be filtered by the upper airway, which is fundamentally different to humans. And so if you're trying to look at deposition of particles in a mouse model, it's probably not a good thing because there's fundamental anatomical and biological differences. That's a crude example, but I suppose it highlights the idea that a mouse is not a human. And so my approach to using these models is to really be very specific about the question that I want to answer and acknowledge the limitations of the model that I'm using. So I think the key in using these systems is to realize that they are a tool, just like a statistical model is a tool for looking at associations. These are a tool to answer specific questions that get you somewhere closer to the truth that you're interested in. And so, as I said, most people think about these models in terms of understanding molecular mechanism and understanding designing new therapeutic targets for diseases and so on, but it's less commonly used in public health. The sort of the most obvious relevance to public health when using these models is basically the field of toxicology, so pure toxicology. And what do I mean by that? Well, in toxicology, and I'm sure you've all seen very similar graphs to this, I mean, you would use them in risk assessment based on population data as well, is the relationship between some exposure and some response. And so you could take an animal model and you might be interested in trying to work out how toxic a particular compound is. So you would expose the system, model, a cell line or whatever, to increase in doses of whatever exposure you're interested in. At some point you get a response, that response increases, and typically you have a dose response curve that looks like this, although they don't always look like this. And you measure the response, you can measure the response in a number of ways. So you could measure, for example, DNA damage. So there you could have cells growing in a dish, you could expose them to some sort of particulate matter or some pollutant, and you could measure the amount of DNA damage that you get. You could also quantify cell death. So you could say, okay, what's the dose of this particular compound that I need that kills a certain percentage of the cells that I'm exposing? You can also use in vivo models. So in vivo models are used to look at mortality. So, for example, you might be interested in the toxicity of a new cancer therapy, and so you would expose an animal model to increasing doses of the compound that you're using to see what dose causes mortality and what threshold you should be using for treatment. Or you could look more broadly at, say, the effects of that exposure on development. So this is a measure of, lost my cursor, a measure of reproductive toxicity. So does that exposure have an effect on development? And there is innumerable numbers of responses that you could use. And so the idea with these dose response curves is you say, okay, well, here's my outcome of interest. This is the relationship between the exposure and the response. And on the basis of that, I'm going to say, okay, well, I'm going to accept a certain level of response, and that's the threshold dose that I'm going to use to make some secondary decision about this compound. You know, how much will you give to a person as if it was a drug? How much would an individual be safely exposed to in an environment? Well, I'll talk about the limitations of that in a second. So this is sort of the low-hanging fruit in terms of the link between the experimental models and how they might relate to public health. So you could imagine that, okay, we've got a new compound, say it's a new cosmetic is I suppose the classic example. What's the relationship between the exposure to this particular cosmetic and the response? Okay. So you would use that information somehow to make some sort of judgment. I suppose the purpose of this talk, or the way that when I was thinking about what I would talk about, is to sort of go beyond this and demonstrate all of the other things that animal models can do in terms, well, not just animal models, but it'll be focused on animals, but experimental models can do in terms of informing public health. So just to illustrate how these sorts of this sort of information is used. If you work in the laboratory space, any of you that have would be familiar with these things. And so these things are material safety data sheets that you get with any compound that you work with in the laboratory. Most people don't read them unless it's a particularly toxic compound. And basically what it gives you is the relationship between the dose of a particular thing and the response. So in this particular example, we have measures of acute toxicity. So if you give this particular compound to rats at this dose, you get an LD50. And what an LD50 is 50% mortality. So if you have a cohort of rats, you expose them to this concentration of the compound orally, then 50% of the rats will die. Likewise, if you use this compound dermally at this dose, 50% of the rabbits will die if you use rabbits as your model, and so on and so forth. And you can see in this, there's some other information. This particular compound is not listed as a carcinogen. It seems to have some reproductive effects. This is a measure of DNA damage. So this is the concentration of that compound that's required to cause DNA damage in fibroblasts, which are a particular cell type. And here's an example where they've also looked at the concentration that's required to kill an invertebrate. So this is a measure of environmental toxicity, if you like. And for those of you who don't know what a daphnia is, that's a daphnia. It's a little water flea. And the reason that particular species is used is it's a sensitive indicator of ecotoxicity. So it's a standardized marker of how toxic a particular compound will be in the environment. And so I suppose the benefit of doing it this way is that you can theoretically do a side-by-side comparison, acknowledging the limitations of looking at animal models of toxicity. But you can do a side-by-side comparison of two compounds. So for example, here's another one, and you can say, okay, well, the data for LD50 in rabbits is this dose for this particular compound, so it's slightly more toxic. Although interest and probably slightly more toxic from an inhalation point of view, although these are very high concentrations. No data on these other outcomes. It's a category 1 carcinogen. It seems to be slightly less toxic to daphnia. And I put these examples up quite often to my undergraduates to talk about the importance of dose response and toxicity and those sorts of things. I'll spare you the going through the process of trying to guess what these compounds are. The one on the left-hand side here is salt, and the one on the right-hand side is the NIST diesel exhaust particle standard. And so the reason I put that up is you can kind of see the problem already. So if you're just going to use animal models for toxicity and you're going to do a side-by-side comparison, these bits of information here aren't particularly helpful from a public health point of view. So they tell you about the acute toxicity and how many animals are going to die, but individuals, if you scale this up to humans, these doses are orders of magnitude higher than anyone would ever be exposed to. So I suppose that's the big limitation and the problem with toxicology as it applies to public health. And so when you're thinking about using toxicological information based on in vivo models to try and make some sort of determination about the toxicity of something, typically rather than using these arbitrary cutoffs, okay, 50% of the animals will die if you expose them to this much, is you look at this at the lower threshold. So what's the dose that's required to see any response? And so this is often called the lowest observable effect level or the no observable effect level. And so you can theoretically use these models, whatever species that you like, to look at the dose that's required before you see any response. Now, again, the problem that you have is that not all systems are equal. So there are published and well-known standards for converting those values into human equivalent. So there are scaling and adjustments that convert this number that you've calculated in your in vivo or in vitro model to the human equivalent dose. And then typically what happens is that there's an arbitrary safety factor that's built into that. So the dose that you end up with is saying, okay, this is our threshold for no observable effect, is often much higher than that, which is perfectly reasonable and a good place to start. But I suppose, again, this highlights the problem that you have with trying to use animal models or cell models to try and predict something or try and use that data to set public health guidelines around exposure to a particular pollutant or whatever, because there is a fundamental difference between non-human biology and human biology. And so if your scaling factors and your adjustments for that aren't accurate, then your data is useless. And my concern in this space is that we get trapped at this idea and that this is all that animal models are good for in terms of informing public health. Does anyone have any questions about that before I move on? So I'm going to change tacks in a minute. This is sort of I suppose an introduction to toxicology and where animal models might sit in that. Stunned silence. All right, I'll continue. Happy to take written questions as well. So if that's our starting point and we say, okay, the experiment models can be used toxicologically, maybe we can use them to set public health guidelines. We need to know what the appropriate scaling factors are. We need to know what the safety factors are and so on and so forth. Okay, so that's kind of the end point of that. The question is, took a while there. Are there other uses for experimental models, at least from the public health point of view? And I would argue yes. And so I'm just going to quickly talk you through a couple of case studies that hopefully provide some insight, or at least give you some idea of my approach to using these models to address public health questions. And so I've lifted this straight out of the NHMRC Ambient Air Quality Standards setting document. There's some names in here I'm sure you'll recognise, Brian Priestley and Michael Abramson. And I've taken this extract from it. It's a really nice document because it kind of integrates, it gives a nice clear summary of how animal studies and in vitro data can be used alongside public health data to set air quality standards. I just want to highlight a couple of points in this statement here. And I think these are the key things. So the critical thing, or the most useful aspect, I think, of these models is your ability to directly show causality and biological plausibility. And so I think that's really key. I don't think they should be I don't think we should hinge or hang all of our, what's the word I'm looking for? Hang all of our hope in these models for determining dose, but what they can do is they can show you, is the relationship you observe epidemiologically, biologically plausible, and is it causal? What they also allow you to do is to unpack the response to complex exposures. So in the sphere that we work in air quality, if you're thinking about the air quality standards, there are air quality standards for particulate matter based on size. You're aware of those. But obviously, not all PM is the same. And so you might have a collection of PM10 from one area and a collection of PM10 from another area. They're all less than 10 microns in size, but they might be fundamentally different from a chemical point of view. So I think the other beauty of animal studies and in vitro studies is you can do side-by-side comparisons of different combinations of chemical exposures within a given framework and compare the responses. So what they allow you to do is to unpack what the most critical component of particulates are, for example, that are most related to the detrimental effects. So what is it about PM10 and PM2.5 that causes the response? Are the responses to PM10 the same, irrespective of the source of that particulate matter? Do they vary? And what are the key components that drive the response? So what I want to do now is to sort of talk through a case study, sort of illustrating how animal models can be used to inform that. And so to do that, I'm just going to start with a bit of background. So I'm originally from WA, so I have a bit of an interest in sort of classic red dust from remote areas and mining towns. And so for some time, I've been working in this space of what I'll call geogenic or earth-derived dusts. And so just by way of a bit of background, there are several communities in the arid region of Australia, not just Australia, around the world, that are exposed to high ambient levels of these particles. Now, we know that there are current air quality standards. One of the problems with those air quality standards is they're based almost entirely on data from urban populations, and urban populations in general aren't necessarily exposed to high concentrations of these particles, whereas remote and rural communities might be exposed to very high concentrations of these particles. Of course, those communities aren't the only ones, and so here's a picture of the 2009 dust storm in Sydney. But just to illustrate that, This is some data that's pulled off the web some time ago. Can't seem to get access to this data now, so which is a shame. But what it showed was real-time particulate monitoring from some of the large mining towns in Western Australia. And so here we have a 1-month period from June in 2012 to July in 2012. The blue line is the PM10 concentration. The 50 microgram per cubic metre cutoff is here, and you can see that over this 1-month period, at the start of during winter, the levels of particles exceeded what the current NEPM standard is several times. And so if you look at this over a year, these communities, they're exposed to incredibly high burdens of particulate matter, but we really have a poor understanding of whether or not what the health consequences of that particulate matter are. Is it the same based on the epidemiological data we have from urban studies? Is it different? Are these particles worse? Are they better? Or whatever. And so I was coming at this from the point of view of, are these particles just as detrimental to health as urban particles? And if so, what are the key components of these particles that cause the most detrimental response? And so without showing you the data, I can tell you that we did do a side-by-side comparison between diesel exhaust particles and these particles. And again, I suppose it highlights the complications. You can't say whether diesel or these particles were worse than the other. All you can say is that the response was different. So we know that if you expose animal models or cells to diesel exhaust particles, the response that you get is fundamentally different from geogenic particles. And so we took the next step and said, okay, well, if we just ignore the comparison between geogenic particles and diesel particles for now, and we just look within geogenic particles, how do the physical and chemical characteristics of those particles vary, and how does that affect the response? I'll skip that slide for a moment. And so just to highlight that point, is if you look at a crude geological map of Western Australia, you can see that the geology varies spatially. And so you could quite easily argue that, well, if you had particles that were blowing off the topsoil here and compared them to particles blowing off the topsoil here, they're going to be chemically, potentially physically different as well. And that's likely to have an impact on the response. So for example, if you took particles from the town of Newman is a mining town surrounded by open cut iron ore mountains basically. And so this town is continuously blanketed, so that's where the data I showed you before was from, in dust that's likely to be very high in iron ore content. In contrast, this is a picture of Kalgoorlie. This is the super pit in Kalgoorlie, the largest open cut gold mine in the southern hemisphere, and here's the town. And clearly the geology here, there's certainly going to be some iron in it, but the chemical composition of particles coming off this space are going to be very different to the particles coming off this space. And so the question is, how does that affect the response? And so what we did was we sampled particles from across the state, and we had 8 particle samples. We measured a bunch of things. So firstly, we measured the elements in the particles, and you can see that in general, and this is true for all the particles that we sampled, they're noting the log scale here, they're dominated by silica, aluminium, and iron. And so we took 8 samples and we quantified the concentrations of these in all 8 samples. We also measured this thing called endotoxin. Endotoxin is a bacterial wall byproduct that's pretty much found everywhere. The reason we have to know that is because those molecules can cause significant inflammatory responses, so we need to know what the concentration of the endotoxin is in our samples. We also measured the MMAD, which is a measure of the particulate size, the geometric standard deviation, which is a measure of size variability, and silica, iron, and aluminium, as I mentioned. So we had these 8 samples, and we had measures of these physical and chemical characteristics. And so what we did is we took those 8 samples and we exposed mice to the particles. Now, I'll try to skip over the details. I won't get too bogged down in this. In short, what you do is you get an adult mouse and you get your particle sample, you suspend it in liquid, you then drop that liquid onto the nose of the mouse and the mouse breathes it in. So we took 100 micrograms of these particles and we exposed each mouse once to the particle and then measured a bunch of outcomes 6 hours, 24 hours, or 7 days after the exposure. And we measured all these things. So the first group of outcomes that we measured were measures of inflammation. So did the mice produce an inflammatory response to the particles? Why is that important? Well, the fact that the mice may or may not produce an inflammatory response to the particles is important for determining the outcome, because if you inhale a particle and there's an inflammatory response, that inflammatory response is being produced to try and kill an invading pathogen. And so if there's no invading pathogen to kill, and there isn't in most cases of particles, then that inflammation was likely to cause damage to the lung tissue. And so that inflammation is then linked to structural changes in the lung, potentially, which will then affect lung function. And so we were interested in measuring the initial inflammatory response to the particles, and then also whether or not this inflammatory response was then linked to structural changes in the lung, which would cause changes in lung function. And it's this that is what causes morbidity in humans. So there's usually an inflammatory response to the exposure, some sort of structural change or immune change potentially that then alters lung function, and that's what makes you have respiratory difficulties. So we measured inflammation and we measured lung function, and I won't go into the details of how we did that. And again, ignoring the detail here, but basically we have the 8 particle samples that we had. Here we have the change in the production of this inflammatory marker compared to control, the change in the numbers of this particular inflammatory cell in the lung, and a measure of a change in lung function. And so I've put these up here basically to illustrate the fact that the magnitude of the response varied depending on what the particles were. So immediately that says, well, not all particles are equal and the response varies in some way. And so what we were then interested in is, well, if that's the case, how do these responses and the differences in these responses relate to the physical and chemical characteristics of the particles? The problem that we had was we had these 8 particle samples, but we had 12 outcomes. And the problem with the 12 outcomes that we measured is they're all highly correlated. And so in order to deal with that, we tried to condense the data that we had on the outcomes into something that we could use to compare between samples. And so the way that we did that is we ran a factor analysis using PCA on the 12 outcome variables. And from that PCA analysis, we were able to identify 2 factors, one that explained inflammation, one that explained lung function. And we used that PCA analysis to then generate scores. So we could score the response on the basis of the inflammatory response and on the lung function response. And what we found is this. This is a summary of a lot of analysis and a lot of data. So basically, in general, there was an inflammatory response, so an increase in inflammation 6 hours post-exposure. By 24 hours, that inflammatory response had gone back to baseline. And interesting from our point of view, and something that I've never really followed up, is there was an overshoot in the inflammation. So 7 days later, the magnitude of the levels of these inflammatory markers that we measured was actually below baseline. So there's some sort of shutdown process where these inflammatory processes are turned off. So there's an acute inflammation that resolves. In contrast, in the lung function measurement, we had this interesting paradoxical improvement in lung function 24 hours after exposure, but 7 days after exposure, there was a persistent decrement in lung function. So we had an acute inflammatory response that may be related to a long-term change in lung function. So we then ran an analysis on the basis of the physical and chemical characteristics that we had to see whether or not the magnitude of these responses was correlated with the physical and chemical characteristics of the particles. And so again, this is cutting out a whole bunch of other things. I just wanted to highlight a few things here. So firstly, what was really interesting to us was noting the negative sign and the beta coefficient for this regression model was that the higher the concentration of silica, the smaller the decrement in lung function. So in some ways, a higher concentration of silica was protected, which is interesting to us because silica is a well-known pro-inflammatory, lung-damaging, pro-fibrotic lung stimulus. And we still haven't unpacked how that might work. What was also interesting is that the concentration of iron in the particles was positively correlated with the response. So the higher the concentration of iron, the bigger the change in lung function. I should also say that the higher the concentration in iron, the bigger the inflammatory response. So the two things seem to be linked. So the more iron that there was in the particle, the bigger the inflammatory response, presumably then the greater the lung damage, therefore the bigger change in lung function. And there was also, and not to discount the physical characteristics, there was also an association between the variability in particle size and the response. So here we started with a specific question. What are the physical and chemical characteristics of geogenic particulate matter that might be related to any inflammatory response or any lung injury that we observe when we expose the mice to the particles? Okay, so we answered a very specific question. We then said, okay, well, that tells us about inflammation and lung damage. What about responses to other exposures? So there is very little, noting that the iron seemed to be one of the key determinants of the response. There's very little data out there on the response, the effect of iron oxide particles, and in fact, most people would argue that iron oxide particles are relatively inert. There is at least some data from places like Port Hedland and Whyalla, where there is iron oxide storage and processing, to suggest that iron oxide exposure is associated with increased risk of hospitalisation for respiratory diseases, in particular hospitalisation for respiratory infections. So on the basis of this observation, we said, okay, is there an association between these particles and the response to a respiratory infection? And so again, this allows, the beauty of the models that we're using allows us to directly test that. So we can say that determine whether or not there's a causal association between exposure to these particles and the response to a respiratory infection. So what we did was we ran a second set of experiments. This time we exposed adult mice again to smaller concentrations of the particles daily for a period of 10 days. And on the 7th day, we exposed the mice to influenza virus. And so this was modelling a situation where you might be exposed to chronic low levels of particles, as you would expect in these towns, to see how this would affect the response to a respiratory virus. And at the end of it, our key outcomes were measures of inflammation again, and also the response to the virus. How much did the virus grow? I'm just going to show you one slide on the data for this. Long story short, the particles, depending on where they were from, increased the viral titer. So the viral titer is a measure of how much the virus is replicating in the lung. So more viral replication is likely to mean that the disease is more severe. And this inflammatory marker here is a critical antiviral defense. And so what we found, again, iron seemed to be the standout in terms of having the detrimental response, is the higher the concentration of iron, the higher the viral titer. And this seemed to be linked to the interferon response. So the higher the concentration of iron, the lower the interferon response. In other words, the lower the antiviral response, which might explain why you see this increase in viral replication, because the antiviral responses are impacted. So now we can see how we're using these in vivo models not to try and assess, not to try and determine what the threshold dose should be, but rather to try and identify the pathways that might be involved and identify whether or not some of the limited epidemiological data we have might be biologically plausible and or causal. And so on the basis of this, we concluded that these particulates cause inflammatory responses, and they also exacerbate the response to respiratory infection, which seems to be positively associated with iron content. Now that's great. As I mentioned before, there's very little data on epidemiological studies looking at respiratory outcomes in these communities. And so now that we had all that information, I thought, well, it would be interesting to go back to the start now and say, okay, could we generate some epidemiological data to support this? And so what we made use of is a survey that's run by the Health Department in WA called the Environmental Health Needs Survey. I'm pretty sure the 2008 survey was done by the WA Health Department, but I can't quite remember. Anyway, so there was an Environmental Health Needs Survey. Basically, it was a questionnaire that was sent out to all of the Aboriginal communities in Western Australia. There are around 300 communities. They received 230 odd responses. And there are a bunch of questions in there about dust levels and about health issues. And acknowledging all the limitations of survey data, we found an association between the levels of dust reported and community concerns about respiratory conditions, including asthma and what was categorised as flu/cold. So again, acknowledging the limitations of the survey data, what we then did was said, okay, can we see any association between community reported dust levels and actual hospitalisation for this data? Hospitalisation data linked to that. And so we used data linkage to look at hospitalisation in Aboriginal children living in these communities. And what we showed was that communities that reported higher levels of dust had increased risk of hospitalisation for respiratory conditions, seemed to be in a dose-dependent manner, increased risk of hospitalisation for ear conditions, primarily otitis media, although the dose response was a little strange in this particular one. And if you unpack that a little bit more, you could see that the response to exposure to self-reported high levels of dust was seen in both upper respiratory tract infections and lower respiratory tract infections. And so I suppose what I'm trying to use this to illustrate is how you can use the in vivo and in vitro data, the experimental model data, to then say, okay, well, we've got this, what appears to be a causal link between two things. How can we then use the that to design epidemiological questions to see if whether or not there's evidence in the public health literature? Sorry, Michael, I missed your question there. The appropriate slide. So H and N are measures of stiffness and lung heterogeneity. So they're measures of peripheral lung mechanics. Okay, so I suppose what I'm trying to illustrate is how you can use in vivo data intertwined with epidemiological data to try and look at public health questions. And so on the basis of the limited epidemiological data on health effects of geogenic PM that existed, we asked the questions, well, does geogenic PM10 cause lung injury? What characteristics are most important? And we addressed that question with an in vivo model. We were then interested in the fact that there was at least some epidemiological data to suggest that exposure to iron oxide was associated with respiratory infections. So we again addressed that with an in vivo model. So does the PM exacerbate infections and what characteristics are most important? So we now had this sort of body of evidence showing that the in vivo models suggest that these particles are detrimental and the characteristics are most important. And we then said, okay, well, could we design an epidemiological study to address that? And so we did an epidemiological study looking for associations between exposure and the health outcome in local populations. And what we're doing currently, so I have a PhD student looking at this, is now that we have that weight of evidence, we've gone back to in vitro models and we're now trying to unpack what the mechanism is. So what are the cells and molecules that are involved in this pathway that would explain these links? And so you can use that to basically generate a weight of evidence data set. And so here we have in vivo models that support causality, while acknowledging the limitations of the epidemiological studies that we have. At least the direction of the effect is the same as what we observe in the animal models, and we can then unpack the mechanisms to try and understand why that may or may not be the case. And so what I suppose I'm trying to illustrate is how you can use in vivo models in conjunction with epidemiological data to answer specific public health questions. That's a good question, Michael. So Michael's asked a question about respiratory pathogens being conveyed by the dust particles. The short answer is yes. So there's some evidence to suggest that the particles can be a vehicle for the pathogens. Although I don't know that there's necessarily data on pathogens, but there certainly is data on allergens, and that they not only act as a vehicle for the allergens, but they can also have an adjuvant effect. And I suspect that's what we're seeing in our model. So because because the exposures are separate, so our geogenic particle exposure and our influenza our influenza virus exposure was separate, I would suspect what we're seeing is almost an adjuvant effect. So the inflammatory response is primed by the particles and you put a secondary insult on top of it. But there's some of these epidemiological associations could certainly be explained by the fact that the geogenic particles are a vehicle for the viruses and the bacteria. And that's something we're certainly interested in. We're certainly interested in the overlap between the ear disease data and the respiratory disease data is, at least in the case of the health of Aboriginal children, one of the key pathogens linked to both respiratory and lung disease is non-typable Haemophilus influenzae. And that particular bacteria, interestingly enough, requires iron, like most bacteria, requires iron to grow. And so that's one of the things that my PhD student is looking at, is whether or not the iron oxide and the iron particles is a bioavailable source of the iron for the bacteria. And so if you have children living in these environments, you've got a situation where they're exposed to the bacteria both via the respiratory tract and by the middle ear, and you've got high burden of particles, and whether or not that high burden of particles is then causing the severe disease we see in the Aboriginal children. I'm quite conscious of time here, Amanda. I'm not sure how long these things typically go for. I've got another case to go. Would you like me to whip through that, or are we better off stopping now and having questions?
Usually we have an hour set aside, and
Okay.
so do you think you would be done within that period?
Yeah, I'll try and whip through it. I think I want to use the next example of starting with really solid epidemiological data and then going to the next bit. So, but what I might do is skip through some of the slides.
Okay.
And so that's an example of a situation where we had limited epidemiological evidence. We used the in vivo models to generate the causality and biological plausibility first, and we then went back to the epidemiological. We then designed epidemiological studies to try and join the two dots. Another way that you can use these in vivo models is to start with a really solid foundation of epidemiology where the mechanism is not known. So, again, lifting another paragraph out of this document, one of the key elements, or one of the key contributions that experimental studies can make, is to this idea of biological plausibility. So you see an epidemiological association which has its limitations, and so the question is that causal? Is there a biologically plausible link? And so we took this approach. This is stuff we did some years ago now, in a study where we were looking at the effects on arsenic in drinking water. And so this is just a slide to highlight that arsenic in drinking water is a huge problem. And so arsenic is a well-known carcinogen. It causes these characteristic skin lesions if you're exposed to high concentration in water. It's been linked to lung cancer, bladder cancer, skin cancer, and a whole range of other, and it's category 1 carcinogen. And this is a map of West Bengal between India and Bangladesh, showing in red dots the shallow tube wells that have unacceptably high levels of arsenic in the drinking water. So there are very large populations around the world exposed to these high levels of arsenic. And so how does, being a respiratory person, you might ask yourself, well, how does this relate to lung disease? Well, there was a really unfortunate exposure event in Chile. So in the 50s and 60s, they tapped into a new water source, and unbeknownst to them at the time, there was incredibly high levels of arsenic. This used to be the WHO guideline for concentrations of arsenic, so you can see it's an order of magnitude higher than that. And there was around 2 decades where the whole population of Antofagasta was exposed to these high levels of arsenic before they did something about it. And so this is a really interesting exposure because it's during a discrete time period. And so there was some really nice epidemiological studies done around this. And there was one published in the late 90s looking at the effects of this exposure with a focus on the risk of mortality from lung diseases in middle age. And so just to walk you through this a bit, so here's the reference population. So this is the rest of the country. And this is the standardized mortality ratio in Antofagasta. The blue bars are people who are exposed in early life. So they were born before this exposure and their exposure occurred postnatally. And you can see that there's an increased risk of lung cancer, which is a well-known effect of arsenic, increased risk of chronic obstructive pulmonary disease, and an increased risk of bronchiectasis. These black bars are individuals who are exposed in utero. And you can see, so they were born, they were in utero during this period. So you can see that the standardized mortality ratio is enormous for these individuals. And just to give you an idea of the scale, so the standardized mortality ratio for smoking sits somewhere around here. So it's almost as potent as smoking. in terms of COPD, incredibly potent in terms of the risk of mortality from bronchiectasis. And so, so the question is, what's the mechanism? And so this is or is there not just what this is mechanism, but is this link causal, and is there a biologically plausible link between these two things that might explain this observation? And so what I'll quickly talk about, so bronchiectasis is basically a structural change in the lung, in the airways in particular, that results from chronic infection. And so the classic examples of this are in cystic fibrosis. So in cystic fibrosis, you have a defect in host defense. And so when you're exposed to a microbial insult, you get a chronic infection. So the infection establishes itself in the airways, it doesn't resolve, and it persists. That chronic infection leads to local inflammation. Because it's chronic, that chronic inflammation leads to lung damage and altered lung structure, and the kind of alterations you get in lung structure perpetuate this cycle. And so you get persistent changes in lung structure that cause a respiratory limitation, and these are irreversible changes in the lung. And so there are basically two broad categories of the defect in host defense that are thought to relate or thought to cause bronchiectasis. And so, sorry, I missed this bit. Bronchiectasis is paradoxical dilation of the large airway and obstruction of the small airway, so it causes respiratory limitation. The two broad categories are mucociliary clearance, and so this is the example in cystic fibrosis. In cystic fibrosis, you have thick mucus lining the airways, and because of that, bacteria are able to persist in it, form biofilms, cause chronic infection, and so on. Alternatively, you might have impaired host immune response. And so the other group of individuals in the Australian context who are particularly susceptible to bronchiectasis are Aboriginal children. So again, this is intertwined a little bit with some of the other things that I'm interested in. And so that's what bronchiectasis is. So here we have exposure to arsenic via drinking water in utero that's causing increased mortality from a respiratory infection-driven lung disease in middle age. And so you can see immediately, so it's a strange disconnect between the exposure and the outcome. And so I became really interested in this, and so we did a bunch of studies trying to look at what the mechanism might be. So again, we went to our animal model, and in this model we used concentrations that were actually well within the range that these individuals were exposed to, and they were exposed for most of gestation. So these are pregnant mice. On day 8 of gestation, they were exposed to either water containing arsenic at a relevant concentration or no arsenic at all. They were born, so the gestation period for a mouse is around 20 days. And then at 2 weeks of age, so 2 weeks of age is sort of early puberty in a mouse, so not quite middle age like the population data, but it's a convenient time point to look at. And we measured lung function and we measured gene expression. So this gives us our functional outcome. So the question is, were we seeing changes in lung function that might be relevant to the epidemiology? And were there changes in the expression of genes that might explain what we saw? So what did we find? So in short, we found changes in lung function in some of the strains of mice. So in this particular strain of mouse, this is a measure of lung volume. This is a measure of the resistance of the airways. And so what you expect to see is as lung volume gets bigger, the airway resistance goes down because the airways get stretched, and so the resistance to flow goes down. Okay, so this negative slope is what you would expect. What we saw is that for a given lung volume, the arsenic exposed mice had a higher airway resistance, suggesting that their airways were smaller. So this kind of fits nicely with the idea of bronchiectasis, which is limitations in the small airways. So this might be one reason why there's an increased risk of bronchiectasis when individuals are exposed in utero to arsenic via drinking water through the mother. So arsenic exposed mice, in utero arsenic exposed mice have impaired airway function. And so then we did some gene expression analysis. Again, I'll skip the details. So we used something called a microarray, which allows you to assess the expression of 28,000 genes on one chip. So I'll skip this too. And so what did we find? So we had these 3 strains of mice. We had this particular strain who had differences in airway function. I didn't show you the data from this strain. This particular strain had changes in peripheral lung function, so their lungs were stiffer. And this particular strain had no change in lung function. And what we found were differences in gene expression in this strain that are known to be associated with cancer. So we know that arsenic exposure increases the risk of lung cancer, so that matches the epidemiology. In this particular strain, which had differences in airway function, we found impacts on a gene related to airway growth, so that makes sense. We also found a gene related to cilia function. So in the airway, there are cilia that beat, that help you clear pathogens. So if you have a defect in cilial function, then that will impair microbial clearance, which might set up a situation for chronic infection. And in the C57 black 6 mice, we found increased mucus production genes. So again, this sits really nicely with what we observe epidemiologically. And all of these things in particular, match the concept that maybe arsenic exposure can directly increase the risk of bronchiectasis by impairing airway growth and increasing mucus production. So this is just a slide showing you what else you can do. So we measured the gene. The problem with measuring the gene is you don't know whether that then correlates with the protein. So you might have an increase in the gene expression, but if that doesn't then correspond to an increase in protein expression, then there's no functional change. So what you can do is you can take thin sections of the mouse lung and you can stain for things. So here we have a control mouse, here we have an arsenic mouse, and I don't know how well it's projecting, but these dark purple cells on the inside here are mucus producing cells. And you can see in the arsenic exposed mice that they have more mucus producing cells. This is one of the genes that is associated with mucus production. Again, you can see low numbers in the airway of the control mice, whereas the arsenic mice, pretty much every cell expresses this mucus producing gene. So you've got to remember that, so this is an exposure again that's happening in utero via the mother through drinking water. These are outcomes we're measuring postnatally, but you can see clear structural changes. So again, when we are able to demonstrate a biologically plausible and causal link between in utero arsenic exposure and the risk of chronic airway disease later in life. And we've quantified that and measured various other things. And so here we have, going back to that slide, here we have a situation where we've shown genes and proteins that are related to mucociliary clearance that might be responsible for this defect in host defense and chronic infection. We've also showed that it can directly affect airway structure. So perhaps the very high risk of bronchiectasis is to do with the fact that you both get structural changes and alterations in mucociliary clearance from the arsenic exposure. Now, I have several slides here talking about the host immune response, which I'm going to skip. And I'm going to go straight to the end here because I'm conscious of time. So, in summary, and so the punchline's been given away, can experimental models be used to answer public health questions? I think the answer is yes. I suppose the problem from my point of view is that when most people think of the link using experimental models to answer public health questions, it's from a pure toxicology point of view. So the link between the exposure and the health outcome, and can you then use that information to determine what the safe dose is? I think that is fraught, particularly for the respiratory system using particulate matter, because the scaling is a huge problem. And there are fundamental differences between lung architecture in a mouse and in a human that, you know, make that link tenuous. Where I think the power of the animal models and the in vitro models is for allowing you to do side-by-side, wide-scale comparisons of complex exposures. So I showed you an in vivo example using the geogenic particles. We've also done something very similar from using house sampled particles and in vitro systems. The advantage of an in vitro system is you can test hundreds of exposures all at once on a high throughput system. So you can then try to unpack what the key characteristics of the particles are that have the most detrimental response. The key to that process is that you then have to make sure that you relate those in vivo data back to the epidemiological data, so you can use in vivo studies to design epidemiological studies. And there's several examples where we've done that. The other advantage of the in vivo and in vitro models is you can use them to demonstrate causality and biological plausibility. And once you've done that, you can then try and unpack mechanism, because in these models you can dissect those molecular and genetic mechanisms because of the tools that we have available. So that's basically all I have to say. Thank you so much for your attention. I should quickly acknowledge the fact that this a lot of the work that I've showed you was funded by the NHMRC and by the CRC, now finished CRC for asthma and airways. I should also acknowledge Dr. Catherine Ramsey, who was the PhD student who did the bulk of the arsenic work, and Dr. Holly Clifford, a postdoc who's still in WA, who did a lot of the geogenic work, and all the other people who worked on the project. Thanks for your attention.
Thank you, Graeme. I think there's a question from Michael.
I think there's another one. How do you measure lung function in tiny mice? Funny you should ask, Michael. Here's a slide I prepared earlier. So this is a picture of the setup that we use. So we anaesthetize the mice. So they're surgically anaesthetized. We put a small tube into their trachea and we mechanically ventilate the mice. What we do is we do two things. Firstly, we have a loudspeaker here that generates a pressure wave. We send that pressure wave down into the mouse's lung, and using the relationship between the pressure wave that we put in and the mechanical response that we get back from the lung, we can use a mathematical model of the lung to calculate airway resistance, which is a measure of the conducting airways, and these two other things which are basically measures of how stiff the peripheral airways are, the peripheral lung is, sorry. The other thing that we can do is, so again, remembering these mice are surgically anaesthetized, is we can put electrodes on their chest wall and we can stimulate an inspiratory effort. And if we stimulate an inspiratory effort, we can then calculate and measure the volume of air that flows in. We can then calculate the volume of air in the lung. So we have these two key outcomes of lung volume, which we know is affected by some of these exposures, and the lung mechanics. So we can really carefully dissect the lung mechanic, the responses in lung mechanics, and where in the lung those responses are occurring. Hopefully that made sense. Are there any other questions? Burning questions?
Yep. Can anyone hear me?
Yes.
Yes.
Hi. Thanks, Graeme. I've just got a question around, you mentioned that currently the air quality standards are based on urban communities. What would you like to see in terms of, would it be a matter of having different air quality standards for each location, or what would you like to see from a policy kind of setting?
Well, potentially. So I should clarify the policy. I don't know if you noticed on that graph that I showed, probably didn't because it was really small. That graph that I showed in the real-time monitoring for the particulates, there's a blue line on it, and that blue line is actually the standard that they use. And that standard is actually higher than the NEPMs. And that was done on the basis of this idea that these particles are inert, therefore they're safer, therefore we can raise the standard. So in those towns, there's Newman, I don't know how broad it is, but I'm pretty sure it's most of the big mining towns in WA, the NEPM standards are relaxed in terms of air monitoring. So from a policy point of view, I would like to join those dots to try and convince people that they're not inert and that at the very least the current NEPMs should be applied. But what we currently lack is whether or not those NEPMs are, as you suggested, are appropriate. And so potentially, at least in these communities, the larger remote communities where air quality is monitored for a specific reason, so they're monitored in those regions quite well, so there's a relatively high density of particulate monitors that are run by the mining companies for that reason. And so theoretically, you could set, from a policy point of view, set air quality guidelines that are lower or higher, particularly in those areas because they're well monitored. And so I think the low-hanging fruit from a policy point of view would be protecting the communities that are in close proximity to mining operations.
Yeah.
Whether you could roll that out more broadly is a separate question, but you know, I think we have enough evidence that these particles are nowhere near as inert as people think that they are.
Yeah. And I have one final question, if I can. with the study with the Indigenous children, you said that it was self-reported levels of dust, but you've just said that mining companies have really good sensors. Is that right? I mean, they collect a lot of data. Do you are you going to be doing some more?
Well, so I
studies
can see Luke's name on there. So I've been talking to Luke about how we might be able to get more accurate data and particulates. The problem with those monitors is, yes, they're well monitored, but most of the Aboriginal communities are hundreds of kilometres from those towns.
Right.
So, yes.
So they're not really the data that you want to be using.
But the one thing about those communities, and you know, I've been to a few of them, and it's a public health tragedy in some ways. Well, it certainly is. The quality of so for example, I think you could reduce the burden of particulate matter in some of those communities simply by paving the main road. And the main road in most of those communities would be 200 metres long. So, because you know, there's traffic in and out of the communities all the time, and they drive straight down the main street, and the houses are about 10 metres from the road. Yeah. And because of the housing quality, the houses are just covered in dust. And yeah, so you know, I've argued that in various grant applications that were never funded. You know, I think there are certainly some simple things that you can do. And, yeah.
Yeah. Okay. Yeah. Cool. Thanks.
You're welcome.
Thank you very much, Graeme. I think in the interest of time and people starting to disappear, we'll close up now. Sure. Thank you very much for that. Next month, Ivan Hannigan should be presenting some work that he's been doing with air pollution and big data. So I'll send out an invite in the next few days. So thank you.
Thanks, Amanda.
Bye.
Bye.